Meeting to talk about PFAS research being conducted in the Great Bay of New Hampshire. This is a hybrid meeting being held at Newington Town Hall and also virtually across Teams. My name is Dr. Jonathan Batali, and I'm the toxicologist with the New Hampshire Department of Environmental Services. And tonight I'm joined by Dr. Paula Mauser from the Department of Civil and Environmental Engineering at University of New Hampshire, Dr. Celia Chen, who's with the Department of Biological Sciences at Dartmouth College, and Dr. Catherine Crawford, who is a professor of environmental studies at Middlebury College. Tonight, we're going to be talking about, again, research topics relative to PFAS in the Great Bay. This includes seafood, aquatic wildlife, and we're going to try to answer as many questions as we can tonight. Next week, we will have a meeting that is going to focus on backyards, garden produce, and more land-based issues. But tonight, we're going to focus in on the Great Bay. There will be technical support provided tonight by Dr. Robert Bissell and Sarah Boyle. They are from my program. They're here to help out in case there's any logistical issues. This meeting is being recorded, and we will try to post it on our YouTube page in a few days. If you have any questions, you can submit them to the chat, or you can use the contact information at the end of this presentation to reach out to us at any time. So again, this meeting tonight is about PFAS in the Great Bay, and we're going to be having this as a recorded session. So if folks want to come back and basically listen to the recording and see what's going on. I realized that we missed a first part. I'll go and try to splice something in later at the office for that. Anyone that's joining virtually, feel free to use the chat function to submit questions, or if you want to hold on to them until the end, we will let you raise your hand, we can unmute you, and you can ask those questions. And for everyone here at the end of this, please feel free to raise your hand. We'll have any time for any questions that folks want to ask. So again, today is really going to be focused on sort of what's being looked at in the Great Bay. So I'm going to be up here to talk about sort of how the state program evaluates risk and how we look at some PFAS issues when we find them in media like sediment or shellfish. Then I'm going to hand it off to Dr. Mauser, who's going to talk about some sources of PFAS in the Great Bay, not only from like the Pease airport area that we think of, but other things that are contributing PFAS to that system. Dr. Celia Chen is going to talk about some of the bioaccumulation studies that they're doing to answer some of the broader questions that a lot of folks are starting to have about PFAS in the Great Bay. And then Dr. Crawford is going to talk about some of the work that we've been doing to actually look at potential exposure and issues around consumption of seafood items and what else we need to understand to really get a handle on risk from PFAS. So we're going to try to answer all the technical questions that we can tonight. But this is a really complicated topic. So in some cases, we might not be the right people. We'll try to identify who the right people are and get your questions to them. And if some of you have those great stump the teacher moments where you ask a question that we don't know or we don't have an answer to, that might help us guide future research projects and things that we need to prioritize or maybe find resources for to address. Now, to start with, I have to do this because I don't know the lay of the entire audience, but I need to introduce what PFAS are. That way, when I spend the rest of this evening saying PFAS, 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 we're all on the same page. PFAS is an acronym for per and polyfluoroalkyl substances. Throughout the talk, we may refer to PFAS as the whole family. There's over 10,000, possibly even more than that, chemicals in this family. But they're basically organic chemicals that have a lot of fluorine on them. Where a lot of us are going to refer to it generally by the family name, we might say a specific thing like PFOA or PFOS, or maybe PFAS instead of PFAS, and then we're just getting into questions about Boston versus Southern accents. So just bear in mind, if we're talking about some of these and acronyms are getting mixed up, we get confused with them all the time too. Ask us to clarify that or understand, right? We're not going to ding anyone for having improper terminology. If you're not sure which one, just say PFAS, and that's the nice catch-all term that we can use for talking about this. Now, the main concern that we have is several of these compounds in this family have been found to be persistent in the environment, and that's because of that fluorine bond that they have. It makes them really handy in industrial uses. It makes them problematic in the environment. They don't really break down, so they move around. And when they move around, some of these can bioaccumulate into wildlife, into us as people, if we are drinking contaminated water or eating contaminated food. And several of these are starting to be determined to be very toxic. Now, I'm not going to have a slide on here going over all the toxic effects of these. There was an ATSDR meeting earlier this week through the CDC talking about some of the research being done on that. I'm happy to answer questions about it, but I want to try to allot time for our other presenters and also to get to the Q&A session. So just understand there is a lot of evolving research around this. We do have concern for the health effects of these. Now, these are examples of some of the ones that we're looking into. This is not the exhaustive list. We look at more than just 4 compounds, but the ability to measure these things is changing and challenging. So some of our presenters might talk about new methods that they're using to look at these chemicals, but they also might give examples of how this has been challenging in some cases to evaluate. How have PFAS been used? A whole lot of places. Oh my gosh, it's a great game of how have you used PFAS in your life. There are a lot of different ways that PFAS have been used for decades now. So I understand the Newington community, there is obviously concern around the Pease former Air Force Base. That's because of the use of a specific type of firefighting foam there. In other places in southern New Hampshire, we have certain textile coating facilities that had industrial emissions. We have a large investigation going on there. But then as we go down this list, here in New Hampshire, we've identified a lot of other sources. A big question for us is certain consumer products and how those contribute to waste systems. But again, this is an evolving list. So as the state is going through and testing and finding more sources, we're finding a lot of places this comes from. Now, to be very clear about risk communication on this list is not up here to be dismissive of any one source over another. I'm trying to make the point that it can be challenging for us to definitively identify which source of PFAS is at a site. In some cases, like where they discharge firefighting foam, it can be pretty obvious. But when we get into some other sources like waste streams, where a lot of this starts mixing together, it can be very challenging. So this is something that as a state, we've been pretty aggressive on here in New Hampshire and trying to tease out. We've put a fair amount of resources into it, but it's still something we're learning about. Now, from a state program perspective, right, I'm a state toxicologist. So what I do is I wait for results to come in from sites where we have sampled for chemicals, not only PFAS. I have a specialty in a whole bunch of other terrifying things. But what we do is we ask how much chemical is there, and then we start trying to evaluate the risk from that. In an ideal scenario, we don't detect anything because then we don't have to worry about it. We can't detect it. A lot of times, though, we do detect chemicals when we go testing for them. And one of the first tools that we use are screening levels. So I'm going to say screening levels over and over again during this meeting, because these are tools that we use to start evaluating, should we continue looking at this? Should we continue investigating this? Does it merit putting resources towards it? And a lot of times these can be customized to different scenarios or different situations. So for those of you very familiar with what's going on at Pease, with the Restoration Advisory Board, the Community Advisory Board, right? They have consultants that are evaluating the site and they are using screening levels there. They're using the screening levels to determine which media should they continue to evaluate, which ones need to keep sort of an eye on in the investigation, or which ones are low enough that based on the information, they don't have to put as many resources towards that. Screening levels vary by site, they vary by region, they vary by state. It really depends on the situation. So these can exist as ranges. I'm going to show you what those are in the next slide. Different from screening levels are actual enforceable criteria or regulatory standards. These are where we have hard stops or we trigger action at sites. They're sometimes cleanup numbers. There's a variety of these different ones. Instead of having a very long exhaustive list here, I'm just going to give one example of the drinking water standard later on. Now, those standards account for other things. They are actual laws or they're enforceable numbers that we use. So they're different than screening levels. So that's just an important thing I try to put out there, because I understand that sometimes when people see that there's an exceedance of a screening level, they ask why there isn't immediate action. And that's because they're different than our regulatory standards. It doesn't mean that we won't act. It just means something's above the screening level. We want to look into it and start investigating. So examples of screening levels relevant to Great Bays. I'm sorry, it's a table of numbers. And I was told in grad school, don't ever do that. It's terrible to do to people, but we're going to do it. It's a table of numbers. So for example, if we look at the top 4 PFAS, there's some ranges of numbers there. for fish consumption in the first column, shellfish in the second column, and sediment in the third column. And that would be in parts per billion units, right? And in those ranges for different scenarios, if you're above that, we'd want to evaluate it more. We want to figure out how much is someone being exposed, really figure out how people are using that area. So for example, if we have a sediment sample that exceeds a screening level, we want to ask, okay, well, how likely is anyone wading in the sediments? Well, if we find out that the sediments are taken from the discharge of the Durham wastewater plant, the only thing that have gone wading in that is me. There was a picture earlier on in the presentation of me crawling through that mud. A different scenario, it's not really something that people are coming in contact with daily. But if it's sediment from an area that we know people are coming in contact with and we can show that they're having frequent interactions, then we want to look into that more. And that's where we work with our site investigators to understand what's the risk, what's the use on the site, what can we do. For things like swimming in water, so that last column is in parts per trillion, that's a order of magnitude change in the units. Those are the ranges that we'd be concerned about people actually swimming, having incidental ingestion, right? So if you're doing like breaststroke or anything, you get some water in, and accounting for potential contact with skin. With PFAS, skin doesn't seem to be the main way it gets in the body, it's really ingestion. So there are some other PFAS that there are screening levels for. I have it just greater than that because the ranges get dramatically higher. And it just started becoming hard to read on the slide if I had all those. If anyone actually wants to know what those numbers are, I'm happy to share those. But also for like PFBS, PFBA, PFHXA, we don't detect those in water or sediments or samples that we've collected anywhere near those ranges. So oftentimes we don't.
That's what you screened and that's what you see.
So that's what we would screen at. So for example, with PFOS, the very top corner up there, if there was fin fish with more than 0.78 parts per billion, we would want to actually ask, okay, how much is in the fish and how much could someone be getting from a serving of eating that fish? So if it's above that screening level, then we might consider issuing an advisory on certain fish, like what we do with mercury or what we do with PCBs or some other contaminants. We might make recommendations around shellfish harvesting sites, so like recreational shellfish where people go clamming. Yeah, so there's a question back there. Mm-hmm. So Dr. Chen is going to actually present some shellfish data later on in this. So we'll just put a pin in that one. So the point is we have screening levels. We do evaluate that data. It's data that when we get it from the Air Force, right, we work with Peter Sand and others to look at that data and understand what's going on. But also you'll see some of the work that we've been doing with Dartmouth and others that we're using to evaluate some of that. So again, these are screening levels. But in other areas, we do have regulatory limits. So for those of you on private wells, you may be familiar with at least the numbers over on the green side, which are the DES, the state of New Hampshire, maximum contaminant limits for PFAS. So these are enforceable numbers that our public water systems have to meet here in New Hampshire, but they have sort of an analogous regulation for groundwater. And oftentimes we use that to screen against people's wells and make recommendations for treatment. Now, full disclosure, the EPA has proposed a new standard. We are currently actually waiting for them to come out with that. They're supposed to come out in December. They're still trying to get it through some review processes internally, but they proposed a lower number, and we're going to see what they come out with in about March or April as the final proposal. So right now they are considering other things. They've explained to us that they may be changing this, they may not be changing it. We can't really act on it until they give us a final number. So EPA is also looking at changing some other regulatory things like CERCLA listing these, which affects waste sites. And they're also trying to come up with ways to deal with this as a family, right? I mentioned earlier, there's likely 10,000 or more chemicals in this family. They're trying to come up with tools or ways that we can start looking at these as a group. It's something that is not finalized at this time. We're still waiting for some guidance on that, but it is something that we're looking for and hopefully having here in the near future. Now, right, the idea is try to keep this high level so I can get on to the other presenters. We do have a lot more information about other investigations that the state has been doing, other things related to standard development, like we're in the process of developing a soil standard, something enforceable. A lot of that information is available on our DES website. You can just Google NHDES and PFAS. It should be the first thing that comes up. It has contact information for program staff that are working in different areas. You can learn about some of our other initiatives, some regulations that are going through related to PFAS. But again, this is more broadly also including other sites in the state. It's not just the Pease Air Force site. It's not just what's going on in Merrimack around the St. Gobain facility. It's a lot that we've learned about through all the work we've done on PFAS. So with that, I'm actually going to hand it over now to Dr. Paula Mauser to present and go over some of the sources and fate of PFAS in these communities. Thank you.
Great. So good evening, everybody. I'm Paula Mauser. I'm here and I'm going to talk a little bit about some of our research and community projects that we've been doing on PFAS over the past 4 or 5 years. So I first just wanted to, should I be here for the camera or can I walk around?
Yeah, you can walk around.
Oh, good. I first just wanted to stop and just give you a little background about environmental engineers. So John talked about toxicology, but I wanted to also mention that my background is in environmental engineering, and so I'm training the next generation of these engineers. And really what we do is we train them to design, manage, and regulate our engineered systems in our communities to control pollution and protect the public health, okay? And so when we're talking about the classes and the research that we do, we work in systems that range from our wastewater systems and the discharges that come from those, our industrial pollution that's producing discharge, stormwater, which is the runoff that's generated when it rains in our communities, solid waste, so solid waste that are going to landfills, so the biosolids from our wastewater facilities that we're managing. We help design systems for air emissions in our industries and municipal systems. And then we're also very involved in cleaning up contaminated soil and groundwater systems. And so the work that I'm talking about today is thinking about these pollution control systems that we design, the infrastructure in our communities, and how we've been thinking about PFAS coming into and out of these facilities, and how we try to understand how we can design them better to reduce pollution into the environment, okay? And I think what's really important to remember is that communities have so many sources and release points of PFAS to our water resources. And so John talked a little bit about some of the specific sources here in the Pease area, but in all of our communities, we have our industrial and our commercial and our residential sectors where we're using all different kinds of these chemicals. And they are basically being conveyed through our underground infrastructure to our wastewater treatment facility. And the wastewater treatment facility is one place where we have a receiver of many different sources that's basically consolidating and concentrating those wastes and then separating them out into a liquid that they discharge into our waterways and a solid waste that we manage either by sending it to a landfill or by post-processing it in a number of ways that can then be beneficially used potentially in a number of ways, or converted to some inert product, okay? And so there's a lot of circularity in how these wastes are moved back and forth, and that's something that we're really concerned about and looking into more specifically. For example, we send a lot of sludge to our landfills, and then the landfills send the leachate back to the wastewater facility. And so if we have PFAS that don't degrade, we can have this back and forth that happens for many years. We can also have air emissions from these different utilities, from industry, from our wastewater systems, from our landfills that contain these compounds as well. But importantly, and this is Durham, where I live, Durham discharges a lot of that liquid, all that liquid out into the river, and that goes from the Oyster River out into the Great Bay. So there's a continuous load of PFAS that's not removed that can be coming into the community from Durham and other locations around us. And so I thought I'd talk a little bit about 3 different projects that we've had going on that relate to some of these different potential sources in our communities and how they interact with our waterways. So first, I wanted to focus in on wastewater facilities. I did a study in 2019 to 2021 where we surveyed 6 wastewater facilities that were in the tributaries that contribute into the Great Bay watershed. And we also looked at PFAS within the Great Bay itself, and we studied how the sources that were coming into the wastewater move through the wastewater system, and then how they are essentially split into what's going out into the water bodies and what's going to the solid phase into the landfills. And so what we see, and this is really pretty consistent, regardless of which facilities that we're looking at, some of them that, for example, have more industrial discharge, like landfill leachate, have higher, slightly higher PFAS coming into them. But generally, the composition of what's coming into the facilities is pretty darn similar. We see about the same number of compounds. We see about the same kind of consistency of the type of compounds. And we see these coming into the influent in the front end, and then they're separated and they become lower in concentration in many ways out into the water bodies, but they're still discharged into our water bodies. But they actually become pretty darn concentrated in the solid phase. And that's actually what we're trying to do, right? We're trying to pull all of those compounds out of the liquid stream, concentrating them into a smaller amount of mass that we can then manage better, okay? So in some cases, some of the larger compounds, the precursors, are concentrating maybe a couple thousand times into the solids. Others of them are concentrating maybe 10 to 20 times, okay? So we have this split, we think about it as fractionation. Does that make sense to everybody? Separate that. We're also doing some work where we're looking at what is coming into our communities just from simple precipitation. So we have some historic air emissions in parts of our state where industries were sending some of their air up into the atmosphere, and those chemicals that remained in that emission interacted and combined into some of our precipitation and then came down in other regions of the state. And this is happening all over the world, which is part of how these are moving globally. But we were really interested in what is actually coming in the precipitation. And the reason why this is super important right now is because we're looking at our drinking water resources and the concentrations that have been proposed for regulation for 6 compounds are very low. And we wanted to know, what are the concentrations that are simply coming into our water bodies just from precipitation alone? Is it possible that just the precipitation could be causing higher levels in some of these drinking water sources? So we set up 5 precipitation samplers in and around the Great Bay Area over the last year. And these precipitation gauges are pretty simple. They have a funnel and a cup, and we collect that rainwater. And luckily, this past year was a really rainy year, so we had lots of opportunity to collect those samples. And we're collecting these at a couple of agricultural areas. So Burley Demerit Farm and the Thompson Farm near UNH, Adams Point, which is down on the Great Bay, and then up in Dover. So these regions represent kind of a rural agricultural up to more of a community developed area, as well as kind of a estuarine to kind of freshwater area. And we're looking at, are there things coming off the farms into that precipitation or in the urban areas or from some of the estuarine areas that we might be capturing in that precipitation that's moving? And I have a student who's actually working on this right now. And what might not surprise you is that we actually detect lots of different compounds just in the precipitation alone. We're finding 8 PFAS that vary in their chain length and their structure, their end structure. couple of them are found in more than half of those precipitation samples, right? So we know that things are moving, right? Atmospherically and coming down into our watersheds. But the concentrations that we're finding in these precipitation samples are very, very low. In many cases, they're close to the detection limits. They're estimated by the laboratories that we're working with. And if we compare the values that we're measuring to the proposed drinking water standards, I can say that we don't see a difference across the different samples in terms of the concentrations. There's no statistical difference. And the values are considerably lower than the proposed drinking water standards. So that's a really good thing. It means that whatever is at least falling from the sky it's probably not going to be a big source in this region of contamination to our waterways. We also compared what we're measuring in the Great Bay region to other types of samples, other similar samples across the world, and found that our system looks really pretty similar in terms of concentration of these compounds to the Great Lakes region. So there's been a nice big study. But there are other areas like China where concentrations are quite a bit higher in the precipitation. Yes, you had a question.
If you're doing this type of analysis, you probably have fingerprints. For example, there's a certain fingerprint of precipitation. There's a different fingerprint for, let's say, a body of water. Are you able to have get any information from comparing fingerprints over time?
Yes, you asked such a great question. That's actually such a great lead-in to my next slide, where we're looking at fingerprints across our communities. So we had the opportunity to work with the city of Dover, and I know that this is a very busy slide, but so that the other studies I mentioned, those were master's students research studies. This was actually a study that was done by our senior capstone group last year as part of their culminating experience. We worked with the city of Dover, who funded sampling across their community of PFAS in many different water media, okay? So the students were responsible with working with me as their advisor and the public works group in the city of Dover, as well as a consultant group that helps out with some of this work for the city of Dover as an advisor. They put together a plan, looked at the potential places that we might be interested in learning more about PFAS in Dover, identified 22 locations that include some of their surface water bodies, their stormwater outfall, so that precipitation runoff running through their urban areas, their wastewater discharge, and then of course the precipitation. And then we analyzed these in the laboratory. And what the students have done is they've actually created a pie chart of the different compounds that are detected across all these different sites. And one of the things that they did was a fingerprint analysis to see how similar the wastewater was, for example, to what we're seeing in some of the other waterways. And interestingly, the wastewater composition, the discharge from the wastewater, which is way down low in the right-hand side, it says WW1 and 2. Those compositions look pretty different than what we're seeing in some of the other places in the community. In fact, one of the things that they identified was that some of the stormwater outfall, so that runoff, into these very shallow areas in the community, they look really different than what's coming into some of our rivers from our wastewater. And they also identified up in the top left, in a surface water body, a composition with lots of a green color. Okay, that's a precursor compound, and it looks completely different from the rest of the samples that we collected. This information is pretty useful to the town of Dover because you can see that regardless of where we looked, we found PFAS in different areas of the community that had slightly different compositions and different concentrations. And it helps them to identify localized areas of concern, especially where they might want to do additional testing. And then also they're thinking about where they're going to put their next drinking water well or source water protection area. And so they can start to investigate what areas, they might want to look at more further as they start that process. So this was a really interesting study. They also overlaid it with information that's publicly available that includes sites of old landfills or old potential sources. And so we can start to overlay some pieces of information to gather why there might be a hit or a different fingerprint in a certain area of the community. So the last piece of research that we're doing right now is to better understand how these compounds are degrading or transforming in the environment and our engineered systems. Now, I know that John said that they're very persistent, and they are, but there are over 10,000 of these compounds, and they have chemical compositions whereby some parts of those compounds can actually break down and generate other compounds. And so as we're looking at this complicated picture, we know that there are polyfluoroalkyl substances, and we call them precursors. And they break down often to intermediate products that are difficult to measure. There's really not a lot of standard analytical techniques for those. And then sometimes they're able to break down all the way to what we call terminal products. And the terminal products are the ones that we actually know quite a lot about, because some of them are regulated in our drinking water systems, like PFOA and PFOS. Those do not break down very readily in the environment. So we have this really big problem where some of the precursors are partially transforming or transforming at different rates in our engineered systems. And I'm showing an example of one that we're studying, and you don't need to know the chemistry of this, but it's called metphos, okay? And it was used in a lot of paper and packaging historically. And you can see that it can break down to many different compounds. And the ones that we measure are highlighted in orange, okay? And so if it's only degrading partway, it can get stuck in certain points. For example, in our wastewater systems, we see that it breaks down a step or two, and then it kind of stays at one of those intermediate compounds for quite a long time before it's capable. But it does eventually degrade to PFOS, which is a regulated compound. So one compound, it can get very complicated if these can partially transform. Yes.
As it breaks down the top of the page down the lower of the page, does the dangerousness, I don't know if that's the right word, lower as it becomes smaller and smaller molecules?
I am not the right person to answer that question. I'm going to pass that to John, but before I do, I will say what does happen is that their physical chemical properties change in the sense that they interact with liquids and solids in different ways. So some of them might become less hydrophobic. They might move out of a solid phase into a water phase more readily, okay?
And what would it take to break them down from larger molecules to small? Is it high temperature? Is it UV? Is it what? Because I imagine, or you have to physically filter it out. Because, for example, in stuff that you're spraying over farmland, eventually, you would love to be able to separate those out. If you could dilute those, or distill those, or burn them, or UV them, whatever it is, what process would it take to get them from a dangerous thing to a safe thing?
These reactions that are shown here are capable of happening under natural conditions, either by microorganisms or by light or by just chemical reactions. But generally, to break these compounds down to mineral pieces, you need really high temperature to do that. So these, some of these reactions happen naturally. Yeah, question.
So you mentioned the highlighted compounds are ones that are being measured.
Yeah.
Does that mean the non-highlighted ones are non-detect, or your analyses just aren't measuring those, aren't looking for those compounds?
The standard analytical methods that the EPA has available would not measure those. You can detect those using non-standard methods. It's a little bit more challenging because in some cases we don't actually have the standards to be able to mark where those are in the analytical tools. But you can find them if you're an an amazing analytical chemist, yes. So, yeah, I know. And so I just wanted to summarize that communities have many PFAS sources coming from industrial, commercial, and domestic sectors. Our engineered systems are collecting and consolidating these, in some cases treating these and removing them, transforming them inadvertently before they're discharged to our waterways or managed as solids. And so our approach is really always source reduction and compound elimination as much as possible to reduce the loads to the environment. And then also, some of our work is looking at the atmospheric deposition also contributes to some of the local pollution. So I'm just going to stop there and pass it over to the next great speaker.
Okay, so thank you, Paula. So our next speaker is Dr. Celia Chen from Dartmouth, and Celia is going to talk about sort of another piece of fate and transport, but really focusing on the work that we've been partnering together on to look at the uptake into aquatic organisms in the Great Bay system. So Celia, are you there?
I am here. Can you hear me?
Yes, we can hear you loud and clear.
Great. Hi,
And
everyone.
just tell me to advance the slide.
Okay. Sorry, I can't be there on the other side of the state here, but I'm an aquatic ecologist at Dartmouth and have been working on contaminants in aquatic food webs for a long time. We worked with mostly on mercury, but more recently we have really pivoted to working on PFAS. We've had the wonderful opportunity to partner with John on a lot of these studies that we're doing. This schematic I really like because I think it gives you a sense of where things are coming from, whether they be the wastewater treatment facilities that that Paula just talked about, or other industrial sources, how it ends up going into groundwater, but also runoff and overland flow. I can't actually, can you see my little arrow there?
No, we cannot.
Okay, that's okay. Anyway, from runoff and overland flow into water bodies. And that's where we know that these contaminants, whether they're mercury, PCBs, or PFAS, do end up in these water systems, water bodies. And in those water bodies, we're really interested in a number of things. Among them, how much ends up in water and how much ends up in the sediments or the mud. We wonder how much ends up being taken up by organisms like fish and other shellfish and invertebrates. And we also wonder whether the contaminants are biomagnified, which means that they increase with each level of the food web or food chain. You might have heard that terminology more frequently. And we know that with PFAS that there is biomagnification of some of the compounds, and not only biomagnification from the lower part of the aquatic food web to higher trophic level fish, but also even into avian predators, like you can see up there in this graphic. And also it does, as Paula mentioned, we know that there's some atmospheric transport. PFAS, we know, has ended up in very remote places like the Arctic and ended up in food webs in those regions. So we know it's not just from areas that have local point sources, and that makes it kind of a problem. We also know that human exposure is in part from water, but largely from food, and we know that in fact from a U.S. study that that high frequency fish consumers have higher serum PFAS concentrations. We also know from the European Food Safety Authority, EFSA, they call it, that fish and other seafoods can account for 86% of dietary exposure. So, you know, knowing how much gets up into the fish and shellfish that we eat is important. Let's see, next slide, please. So we've been working on a number of studies. The one that this map shows is one that I've collaborated with Kate, Dr. Crawford, who's going to speak in a moment. We had a grant from the National Institute of Health to look at PFAS in shellfish as well as in demersal fish. I'm just going to show you some of the data from the shellfish study. What we did was we had 7 sites around Great Bay, and as you can see, the pink area there is the former Pease Air Force Base. So you can see where our sites are located, and the names: Boston Harbor, Fox Point, Hilton Park, Odiorn Point, Wentworth Mansion, at Route 1A, and Woodman Point. Woodman Point is the one that was closest to the Pease Air Force Base. And we collected 5 different bivalve species across those sites, including softshell clams, blue mussels, European oysters, American oysters, and razor clams. We also took surface water samples, and we also took sediment samples. So in our world, we're really interested in knowing, well, do the different kinds of, we have so many PFAS compounds to choose from, do they all partition or end up in the same place? Do they end up in water? The same ones end up in water as the ones that end up in sediments? And does that matter to the way that different organisms feed? So some of these organisms, they live in the mud. If any of you have been clamming, you know you have to dig for some of these shellfish. Others, like blue mussels, are usually suspended and filtering their food from the water column, as do oysters. So each of those different kinds of shellfish have different, what we call feeding strategies. So trying to understand what's in water versus sediment is also important to determining, well, what kinds of compounds end up in what kinds of organisms. Okay, next slide, please. So let me orient you, and I can't do it with an arrow. So on the left side is a graph, and it shows on the y-axis, the vertical axis, the percent composition of the total concentration of PFAS. So the different colors on those bars actually refer to the compounds that are shown on the right side of the graph that's on the left side of the slide. And so what you can see is that there are different colors. And just to orient you, so Paula talked about different chain lengths of PFAS, and she also mentioned that there are carboxylates and there are sulfonates. And so if I start from the top 2 chemicals on the right side of that graph, the phosas are the precursors that she referred to earlier, and those 2 blue-colored bands are ones that could end up transforming into PFOS. Then the next, PFDS, PFOS, the brown to tan colors are the sulfonates. They all end in S, you can see. And those are also going from longer chain to shorter chain. I hope this isn't too confusing. And then below that, we have the green down to the yellow, and those are carboxylates, and they are also going from long chain down to shortest chain. So looking at the graph then, what you can see, and across the x-axis, the horizontal axis, you can see the different kinds of shellfish. And the last 2 columns are water and sediment. And what you can see, I think, pretty clearly is that, first off, blue mussels and oysters and softshell clams have most of their PFAS as the green and the brown. So they have some they have some carboxylates, especially long chain, and they have some sulfonates, also longer chain. But then when we look at razor clams, it's kind of different. And we have some shorter chained carboxylates, the yellow, but we also have some of the longer chain carboxylates, and we have PFAS, which is the longer chain sulfonate. And then when we look then at the final two, we see that water and sediment look really different from the bivalves. And what that suggests is that even though some of these, like softshell clams and razor clams, are literally living in the sediments, they do not concentrate all the compounds that are in the sediments. They actually, those two columns, the clams versus the sediment, look very different. And the sediment and the water two bars look very different from one another as well. And finally, we see in comparison between oysters and clams and mussels as compared to razor clams, the razor clams have very different composition in terms of the percent of different compounds. Okay, turning to the right graph. The right graph really shows you the concentrations in each. So what I haven't shown you, and I didn't give you a very good key for, is the BM stands for blue mussels on the y-axis across the horizontal. Oysters are OY. RC is razor clams. SC is softshell clams. And what you can see from this, I think pretty clearly, is that all of the blue mussels, oysters, and softshell clams are pretty low. But the razor clam is really high in concentration. So this is PFAS, okay? This is just PFAS. And the reason that we show you PFAS here is because that is one of the compounds that has screening levels. And they are on the right side. And you can see that the PFAS in razor clams exceeds the New Hampshire and EPA screening levels. And in some of the samples for adults, and all of the samples for, most of the samples for children. So that was really surprising to us, because what is it with razor clams? And we don't really know yet. We have some more samples that we're going to run, but this was, and we also don't have across all our sites all the species. So trying to determine, well, is one site higher than another? We can't really make those comparisons because we don't have them across all species across all sites. But we do know that PFAS is very prominent in every sample type, and that the biota are dominated by the longer chain carboxylates and sulfonates. We also know that the water and sediment concentrations are really low. I mean, so many of our samples were below detection for water and for sediments. And we didn't see a gradient of higher concentrations in water or sediments closer to the Pease Air Force Base, former Pease Air Force Base. So that was kind of a question we wondered about, and we don't see a pattern. And then finally, the razor clams are super high compared to the other taxa. And this is these data are currently being worked on by my technician, Christine Gardner, who is writing this up, and with John as well, to hopefully submit as a manuscript soon. Okay, next. So that was our first field study in Great Bay. We then got a grant from New Hampshire Sea Grant, as well as from the DoD, Department of Defense, to look at further the bioaccumulation of PFAS into food webs. So here I'm showing you a map of the next study, where we went out in summer 2022 and sampled 10 sites. And the 10 sites included 3 sites at Dover, Durham, and Hampton. That's sites 8, 9, and 10 on the map, just at the outfalls of the sewage treatment plants. And then you can see the other sites, numbers 1 through 7, are again at varying distances from the Pease Air Force, a former Pease Air Force base. Chapman's Landing is a little bit farther away, number 2. But what we tried to do then is to sample at all of these sites for the same taxa, because as I said before, oysters we didn't have the same taxa in the shellfish study. So next slide, I think. Yes. And so here we collected water samples, sediment samples. We actually also measured things like salinity, conductivity, a whole bunch of other parameters. But we also used fish staining and trapping and invertebrate collections to gather as many of the pictured organisms on the right: mud snails, periwinkles, rib mussels, blue mussels, green crabs, the clams and oysters that we sampled before. And we also collected killifish, or it's also called mummy chog, at all the sites. We plan, and we have been processing all these samples, and they are now pretty much ready for extraction. We also collected samples for stable isotopes, which we use to determine where these organisms are getting their food source. So for instance, it can decipher whether animals are getting it from the water column or from the sediments. So that's another thing we'd like to know, because in fact, if they're getting them from the sediments, what are the sediment profiles? I think someone earlier asked the question about fingerprints. Well, we're wondering whether the fingerprints in the benthic feeding organisms, the ones living and eating in the mud, look similar to the sediments. We saw that they didn't so much before in the earlier study, but now we have a broader range of organisms. And I think that's, yeah. And so anyway, those samples are right now in our lab about to be analyzed. All of our samples are actually we extract them in my lab, and we send them over to Clarkson University, where our collaborators, Tom Holson and Sujan Fernando, have an analytical lab for measuring 40 PFAS compounds using the same 1633 method that Paula had mentioned. So anyway, I don't have a summary slide, but that's where we are with our research, and love to talk about more of this later. Thanks.
Okay. And then I think this is just a slide of some of the data from
Celia
had presented earlier, where this is just the individual samples for the different sites. So for the sediment, you see it's below 0.2 parts per billion. For water, below about 8 parts per trillion combined. So this is just another summary slide of that. We will be posting the slides. We'll make those available online. I'll be working with Martha to get that up and accessible for folks that want to see this in closer detail, because I realize on the screen, some of the small acronyms are probably straining folks' eyes at about pushing 7 p.m. So yes. With that, I'm going to thank Celia so much and have her stick around for Q&A at the end. And then we're going to hand it over to Dr. Crawford now. Kate, are you with us?
Hi, can you hear me?
Yes, I can. This feels a lot like a seance sometimes. So thank you for being here.
Wonderful. Well, thanks for having me. And again, I apologize I'm not able to be there in person, but calling in from Vermont, and it's wonderful to be able to join you all tonight. And thanks for being excited to hear some of our research. And we're, I think, as all of the other presenters have mentioned, also really looking forward to kind of hearing about how community interests might be able to inform our research directions moving forward too, since I think all of us value that part of the hopefully bidirectional work that we're able to do in our research programs. So I'm an environmental health scientist by training, since we seem to all be introducing ourselves and our backgrounds in that way. And from the perspective of an environmental health scientist, we're interested in answering questions, broadly speaking, about how concentrations of chemicals in the environment ultimately end up potentially impacting human health. So we're thinking about concentrations of things that are in the environment, what is in the environment in the first place, how people might be exposed to those compounds. Actually, they're not even always compounds, right? Sometimes environmental health scientists study things like climate change. So environmental factors, broadly speaking, and how those conditions might end up impacting human health, so how people are exposed to them, and how those exposures then can alter physiology, leading to disease, or in some cases, hopefully not disease as well. So with that context in mind, I should also mention that I'm an assistant professor at Middlebury College, so I do a lot of work with undergrads as well. I don't have any conflicts to disclose for this talk, and I also wanted to mention that this research, as a lot of the work that Celia and, Dr. Chen and potentially Dr. Mauser as well had presented, is unpublished, so I do ask that you don't copy or distribute it, but I wanted to be able to have a chance to share it with you since, again, hopefully you all will find it interesting and potentially have some great suggestions for how to advance it moving forward. So as has been indicated by other information you've heard so far today, there are a lot of different sources of PFAS in the environment, and that then informs the need to have regional data to understand concentrations in environmental media, because there is the spatial heterogeneity that we see across the landscape, or this difference in concentrations and profiles that we see in different places. And so having that information about a localized or regionalized condition is really important to then developing protective measures for common routes of exposure. As has been mentioned, we generally consider PFAS or people to primarily be exposed to PFAS through ingestion. So that includes drinking water as well as diet, and that seafood can for some people be a really significant source of PFAS to them via diet. We also know that we need kind of more robust data about commercially harvested seafood. So as Dr. Chen was just sharing with you, there's a lot of information, I shouldn't say a lot, there's a growing body of information about seafood, broadly speaking, in freshwater and marine environments, but that's all well and good, but knowing what might be on people's plates, especially people who who consume seafood that they've purchased at a retail market, is a really important part of the equation for understanding human health risk. So this particular study is trying to address that gap of what is actually in seafood purchased at a retail establishment. So next slide, please. Great. So we began this work by conducting a seafood consumption survey amongst New Hampshire residents, and the survey was developed based on EPA recommendations for best practices for developing robust seafood consumption surveys, as well as a relatively recent Idaho fish consumption survey. We then partnered with the UNH Survey Center to leverage their existing Granite State panel, which is a pool of potential survey participants that has been specifically recruited based on random cell phone and landline numbers to be representative of the state of New Hampshire's population. Our survey launched in June of 2021, and we had roughly 1,800 respondents, and 163 of those had at least one child ages 2 to 11 years old. I'll mention now that we specifically looked at this age range because the US EPA and FDA, Food and Drug Administration, seafood consumption guidance specifically provides recommendations or focuses on recommendations for seafood consumption amongst young children, so this age range under 11 years old, and also women who are either pregnant or might become pregnant, because those two populations are particularly susceptible to the toxic effects of PFAS given the small body size of those individuals. Next slide, please. So our survey was an online survey that took about 10 to 15 minutes and asked people a range of questions about sociodemographic characteristics, as listed here: age, county of residence, race, ethnicity, income, etc., as well as information about their seafood consumption habits, so that we would be able to calculate an estimate of daily seafood intake. In order to do that, we needed to know information, for example, about how frequently people are consuming fish, as well as the portion sizes that people might be consuming. As you can imagine, it's pretty hard to recall how many ounces of seafood you consumed when you might have not eaten seafood since last month. So we used images like this in our survey to help people more accurately report their seafood consumption patterns. Next slide. Great, thanks. So then the other half of this equation, right, was understanding what PFAS is, or what concentrations of PFAS are present in what we call market basket seafood. And market basket in this case is just kind of a jargon term within the scientific community here to think about commercially harvested and commercially purchased seafood. So in this particular case, we were looking at these 7 different species of seafood: cod, haddock, lobster, salmon, scallops, shrimp, and tuna, that were purchased at a seafood market in Portsmouth, New Hampshire in May of 2022. And specifically, we measured 26 PFAS compounds in 3 samples of each of these species. And we'll come back to some of the details about the sampling in a few slides, but I'll leave it here for now. So we then used several statistical approaches to, again, relate this information about how much seafood people are actually consuming, and also the PFAS concentrations in that seafood, so that we could then start to look at risks that people might be experiencing from consuming that seafood. So specifically with using the survey data, we calculated descriptive statistics, so kind of general information about the mean or the average person, as well as the range of seafood consumption patterns. And I should note that this particular survey data was weighted based on age, sex, education, region of the state of New Hampshire, and political party, which is how that Granite State panel that I mentioned before was designed by the UNH Survey Center. So all results that we're reporting are weighted based on these demographic characteristics. We also then calculated, summary statistics for all of the 26 compounds in each of the species of seafood that we had data for, and then ultimately ended up calculating this compound-specific estimate of daily exposure or ingestion of that PFAS compound. And we did this for a few different scenarios. So the median and maximum concentration of PFAS in those seafood tissues, and also the median and high frequency seafood consumers, so that we would have a bit of an understanding about the range of types of ingestion or exposure levels that people might be experiencing, something that would represent kind of the average or typical person, and then also that highly exposed individual. And the reason that we do that is when we establish things like fish consumption advisories or think about exposure risk from consumption, we want to make sure that our kind of assessment of that problem is not just the average person, right? We want to make sure that we're also being protective of the person who might, for a number of reasons, consume more seafood than average. And we want to make sure that that person is also accounted for and protected within these equations. So we then took those scenarios for the daily ingestion rate of the various different PFAS compounds from our seafood that we looked at, and we ended up comparing these to what we call health guidance values. And these are values that are set by typically various government bodies that are used to, or they're designed basically to be health protective, or to be a level that is not likely to be associated with any excess risk if people are to be consuming seafood, for example, on a daily basis. And so we're able to compare what our daily rate was that we calculated based on our conditions to those health guidance values, to basically say, are people consuming seafood in the state of New Hampshire at the rates reported in the survey more or less than what would be considered to be a level that would be potentially causing excess risk in that person. So next slide, please. And apologies if that's convoluted. Happy to answer questions later, but we'll move on for now. So our seafood consumption survey indicated that New Hampshire residents tend to consume relatively high amounts of seafood. You can see here that for adults, most people, so 94% of people, had consumed seafood within the past 12 months. And roughly half of those adults also indicated that they consume seafood more than once a week. Slightly fewer children consume seafood, but still relatively frequent seafood consumers within the state of New Hampshire. I also want to point out that not too many people, roughly only about 1 to 3% of individuals reported consuming any self-harvested fish, indicating that, again, the vast majority of these people are consuming seafood that they've purchased commercially, whether at something like a seafood market or grocery store and then prepared at home, or at a restaurant where they might have had someone else prepare it for them. Next slide, please. Great. So then we were also, of course, interested in portion size. How much are people eating when they do go about consuming seafood? And we see that adults typically consume portions of around 4 to 6 ounces. So if you can remember from that picture before, that's roughly the size of the palm of your hand. And children tend to consume lower portion sizes, not surprisingly. Then we were able to calculate these daily consumption rates. So the median, or that kind of typical or average daily consumption rate for adults is 33, from our study was 33.9 grams of seafood per day. And for children, it was 5 grams per day. I want to call attention in the bubble on the right here to the fact that this is higher than NHANES, which is the National Health and Nutrition Examination Survey, reports for Northeast residents. In that particular survey, they've found in the past that adults tend to consume about 23.9 grams of seafood per day. So New Hampshire residents do tend to, based on our survey, New Hampshire residents do seem to consume more seafood than has previously been reported. And we can answer any questions about that if they come up at the end. Next slide, please. So then we wanted to know something about what species are people consuming in the first place. And so here we have figures for both adults and children. I know it's a bit hard to read, but I will just say that shrimp, haddock, salmon, tuna, scallops, cod, lobster, etc. The species that I had mentioned before and are highlighted here with these red stars are the ones that were most frequently consumed by both adults and children within our population. Next slide, please. So then again, we took this information and we coupled it with the PFAS concentrations that we detected in these market basket seafood. And I want to take a second to walk you through this figure here. So on the y-axis, we have concentrations of the various PFAS compounds reported in nanogram of PFAS per gram of tissue. And on the on the x-axis, we have the different PFAS compounds. And you'll notice that we don't report all 26 compounds here. That's because we've only reported the compounds for which there was at least one tissue sample with a level detected above what we call the limit of detection for those samples. Another thing to note here is that there are 3 different kind of symbols that are listed. There's a square, a circle, and a triangle. Each of those represent one of the 3 samples for that species that we looked at. And the closed circles are concentrations that were detected above the limit of detection, and the open symbols are those that were not detected above the limit of detection or below the limit of detection. You'll note here that we are only reporting information on shrimp and lobster, and that's because these were the 2 species that had the most number of PFAS compounds detected in them and also some of the highest concentrations, and also were species that were frequently consumed in our population. So that's how we arrived at focusing on shrimp and lobster for this paper. What you'll see here is that there's a range of different PFAS concentrations detected, but all of these compounds tend to have pretty high detection frequencies within these particular species. Next slide, please. So again, the real question, right, was how do we integrate seafood consumption patterns and these PFAS concentrations that we see in these seafood tissues, and then estimate whether the amount that people are being consumed is potentially problematic or not. And so we did that, one, by taking the daily PFAS dose, which we calculated based on body weight that people had reported in the seafood consumption survey, as well as seafood consumption rate. So again, that amount of seafood that people are consuming per day, and again, that kind of median or typical amount, as well as that high amount, and then also the PFAS concentrations, looking at both the median concentration as well as the maximum concentration for each compound. Then we evaluated this, we compared this to existing health guidance values. And these health guidance values, again, as I mentioned, are derived by various government entities to be representative of a concentration that would not be expected to cause appreciable risk to that individual if consumed on a regular basis. And so the 3 compounds that there were health guidance values available for us to compare to were PFNA, PFOS, and PFUNDA, and these are the health guidance values as well as the entities that had developed them in the first place. So we were basically comparing the number 1, the daily dose, to number 2, that health risk or that health guidance value. And then number 3, great, thanks. What we found was that based on our data from this study, PFNA did not present excess risk at a level that was concerning under any of the scenarios that we considered. So again, that median and high seafood consumption and the median and maximum PFAS concentrations. However, for shrimp, high frequency or high consumers at the maximum PFAS concentration were associated with a risk that did exceed the health guidance values for both adults and children. And for lobsters, again, considering that high seafood consumer, both the median and maximum PFUNDA concentrations were associated with a risk above the health guidance values for adults and children. And so the kind of main conclusions for this paper were that, one, importantly, New Hampshire residents do seem to consume more seafood than other regions of the US, and including previous results for the Northeast specifically. People do tend to consume commercially harvested seafood that's readily available in grocery stores most commonly. And based on the information that we have from the study, PFAS and PFUNDA may present risk of exposure amongst high frequency seafood consumers and shrimp and lobster, respectively. There's a lot of work that needs to kind of continue to go into this body of research, because it's one that, as indicated in that very first slide where I was identifying some gaps, we know that there's just not a huge amount of data out there on commercially available seafood. But based on that information, we know that we need to continue evaluating this based on the beneficial and competing negative risks of seafood consumption, both from the contaminant exposure level and the nutritional benefits perspective. And we also need to make sure that our work continues to consider people who may have seafood as a really particularly important dietary staple, either for cultural reasons or other preferences, which, based on the way our survey was conducted, are not kind of preferentially considered in this study in any way. So with that, there's a whole host of people who have made this work possible, and look forward to discussion and taking any questions as we move forward.
Great. Thank you, Kate. So with that, we're actually to the end. So if everyone's like adjusting their seat and see if their back cracks. This is our contact information. I've also included the contact information for Peter Sandin, who is here tonight. Wave, Peter. Because I understand there are probably going to be some questions that people want to ask specifically about the PEAS investigation. And this is not a RAB or CAD meeting tonight. We may be able to answer some questions, but some of those we may just have to take back to those appropriate meetings. But Peter is here to answer specific questions, or he can take those questions if you want to take his contact information and follow up. I've also included Amy Russo, who is not here tonight because, one, she has a lot of other engagements, and two, I think one of her other kids is sick again. They just keep going back and forth giving each other something. But she's actually our PFAS response coordinator for the department, and she oversees a lot of the programs that either direct funding or help organize well testing, or a lot of other things that we're working on in the agency. We have Dr. Ballister's contact information, Dr. Chen's contact information, Dr. Crawford, and then I also have a whole slew of links here to be accessible that can connect you to things like the EPA's website, information from the CDC. We also have a New Hampshire specific fact sheet about other PFAS exposure sources and just some other useful links for folks. So with that, we'll go ahead and open it up for questions and discussion. And we have a hand up over here. Dr. Crawford, did you hear that?
No, I don't think we can hear the audience particularly well, so you may need to repeat questions.
Right, so I'll repeat the questions. Okay. So the question was, in your work, did you compare farm raised to wild caught fish, or was it just specifically grocery store fish? What was sort of the categorization of that?
Yeah, that's a great question. In this particular study, we were just looking at wild caught seafood. So this was seafood that was purchased at a commercial seafood market in in Portsmouth, New Hampshire. And this was, for the most part, all seafood that would have been sourced from around the Gulf of Maine and brought to market that same day. Dr. Chen, if I'm mischaracterizing that by any means, please hop in, but that's my response to that question.
Yeah, I would say that we do think that they could have been sourced around the Gulf of Maine. We don't know for sure. We didn't, we just purchased it. We did not ask the seafood store where they had gotten their seafood. And we also handled it in the way you would with food wrapping and all of that, fish wrapping, in the same way that you would if you went to buy that fish, that seafood.
Yes, thank you. That's a good point. We were really wanting to represent what someone would actually be exposed to. So we certainly did not try to exclude any packaging that could possibly have been a source of seafood, but that wasn't necessarily going to be useful to this particular research question. So we included that in the study.
Yeah. Okay, we have a second hand up over there.
At relatively high concentrations. I see quite a few neighbors that have lobster traps out in Great Bay. And there's also, you can get lobster out in the wide ocean. Is there a difference that we know of in how much concentration of lobsters that are caught in the bay versus outside of the bay?
So Dr. Chen, the question was, do we know the difference in PFAS concentration in lobsters caught within the bay or outside of Great Bay?
Yeah, that's a great question. And I actually just took on a PhD student this past fall who is really going to dig into that. And we've learned just from reading papers recently that there is a resident and a transient population of lobsters that that are commercially as well as recreationally harvested. So no, I think it's a very good question. We've been wondering about lobsters, not just because of this study, but the state of Maine has also published in a recent report data on the PFAS profiles and concentrations in Maine lobsters. So, and they're finding them, and there's about 5 compounds that are fairly consistent across all the sites in Maine. So it's a very interesting area, and yeah, we hope to know more about that later.
Let's go here, and then we'll come over to.
So about finfish, like saltwater finfish, I know a lot of species that are recreationally, commercially harvested here and consumed or migratory, and so you might not expect levels increased from contamination in Great Bay, for instance. Has there been any effort to look into the levels of migratory finfish?
So Dr. Chen,
yeah, I can hear that one. I don't.
Yeah, okay.
I don't know why. Some people we can hear. Some people we can't hear at all.
Okay.
Yeah. So as part of that study that was on the shellfish, we also had a sample set of demersal fish from the Gulf of Maine. So they aren't transient species going in and out of Great Bay, and I didn't mention it because but we are seeing some compounds appear to be higher inshore than offshore within the same species. And we also see that, in fact, it's kind of interesting to us anyway, that there's some long chain PFAS in those demersal fish that, are higher than PFOS, for which we know there's a screening level, but there are other compounds that are longer chain than PFOS that are showing up in these marine fish. And they're showing up in lobsters too, by the way, and shellfish. We know a little bit about fish. We haven't been sampling any of the recreationally caught fish just in Great Bay, but that's a really good question. Possibly we should, but that wasn't part of our study design. Yeah, good thought though.
I will add on to that. One of the challenges that we've run into as far as sampling, and I think Dr. Chen, Dr. Mauser, Dr. Crawford can attest to this, is the cost of doing these studies is pretty high, and we have to be really selective with using the resources for whatever samples we have access to. So a lot of these demersal fish or these ocean fish that Dr. Chen is referring to, several of those were actually caught in a previous study that her group has been working on with mercury, and they were able to make advantage of samples that they already had. Several of these projects have also built on some seed money that DES was able to provide when we had a one-time infusion of funding from the legislature. Unfortunately, their group has been able to build on that and bring in additional resources, but this does continue to be a challenge.
So it would probably be simple enough to get a recreational or commercial fishermen to give samples, if that works with your protocol.
Yeah, and I believe, Dr. Chen, that's how you got your demersal fish and your offshore fish, was finding
Yeah.
a friendly fisher person to help out.
Well, when we wrote that grant, we actually put, well, the original grant that was for metals, actually, we actually put a budget in for hiring David Gay. I don't know if any of you, not David Gay, sorry. One of the local fishermen out of Portsmouth, a New Hampshire fisherman, and he happens to be very interested in the science around, these commercially caught fish, because actually it's not that easy to get commercial fisher people to want to get samples for contaminants. But anyway, he was helpful. And we also got samples from the National Marine Fisheries Service in Woods Hole. We made a request they do a demersal fish survey every 6 months, and we made a request for fish from them as well. So yeah, we've tried, we're always concerned about, budgets really. And so we were able to leverage those fish samples.
Andrew?
I had a couple questions. I was curious if all of the seafood that was purchased at the Portsmouth market, was it all on the same day? Like all the samples purchased at the same time, and would we expect maybe different times of the year we could see fluctuating levels for various reasons?
Did you all hear that question?
Yes, it was all purchased on the same day. The seafood market guy, we had to tell him we needed 3 replicates from 3 different fish, which he thought was rather odd. And in terms of seasonal differences, I think that's a great question. I mean, certainly in, I know more about mercury. I don't think we know this for PFAS at this point with fish populations, but say we know it with mercury because fish don't feed as much in the wintertime, and so their body condition declines. Sometimes in the wintertime, for mercury anyway, their concentrations actually increase because they're not putting as much body mass on as they are taking up, the contaminant, in that case, mercury. So I think it is very possible that there could be seasonal differences in a population of fish species. But we have not included that in our studies so far, partly because of what John just said about how much it costs to run these samples. But it's a great question, and it is possible that they could be different.
Other?
Yeah, I was just also curious if for Dr. Crawford, it seemed like the data focused on the lobster and the shrimp, but will the additional sampling from the other types of species be part of the paper? Like, will that data be made publicly available at some point?
Yes, in the supplemental tables for that manuscript, which again is not published yet, but hopefully it will be before too long. There's additional information about concentrations of PFAS in the other species. Again, as you indicated and accurately captured from my presentation, these were the ones that we kind of focused on for the main feature of the study, but you will be able to find those additional values in that paper.
Are there other questions?
Just curious, what would a person's political standard affect in the survey? I can understand your age, your sex, everything like that, but your politics?
So when the UNH Survey Center asks that, they ask it to make sure that they're getting a whole representation of the New Hampshire population. It doesn't affect the fish consumption piece. They try to make sure that when they're reaching out and polling everyone, it's representative of everyone across the state.
And Dr. Patali, I might add too that we do know from research that there are differences in typical response rates to the things like surveys, particularly for academic research participation across political parties. So I think that primarily the answer is exactly what Dr. Patali just indicated, which is that it's kind of the way that the survey was, or the Granite State panel is designed, but it is also just an important consideration to think about too.
I have a few more questions if everyone else. I wanted to know if there is enough data to support an advisory on razor clams based on what Dr. Chen presented with the PFOS levels being high in those.
So we're working with Chris Nash in the shellfish program to evaluate that. Some of the sites that we sampled from are already closed for other reasons. But one of the things we're trying to understand, as Dr. Chen mentioned, why is it that razor clams are that high? Her group does have other samples that we're waiting to get those analyzed, but we're also working with Chris Nash at the shellfish program to see if there's other sites to go out and collect those from. The trick too is razor clams are actually pretty rare to find, and that's one of the reasons where he's saying we can probably find some sites to do some more analysis, but we want to have just a little bit more data before we issue out something specific to all razor clams. We want to make sure it's specific to that site if that's the case.
We also have some razor clams from the 2022 sampling. And yeah, those were just, there weren't that many replicate samples from that. So I think we do need to get more data just to see if it's consistent.
Another one of my questions was about the July 2022 sampling that you did like in Hampton, and like when will that data be publicly available?
Oh, yeah. Well, we don't even have the data yet. So this 1633 method, which we have to use to analyze our samples, has really held us up. We have had to go through all kinds of standard operating procedure reviews about the method and just things like that. Anyway, and so that's why it's a 2022 sampling and they're in the queue for extraction. There's been a lot of methodological holdups for this. Gosh, I wish I could say, I mean, I would say, I hope that within the year we would have something. Yeah, things have been taking longer than we had hoped.
Yep. So really quick, we had a question.
Well, just why do we have all this PFAS in our environment, and how can we, are we still pouring PFAS into our environment, and can we not get the EPA to stop letting people produce it?
I can take a stab at the broader regulatory quandary that we're in. Paula, I don't know if you want to start with just the source issue.
We just have so many different products, commercial products that we use on a regular basis that contain these compounds that continue to flow into our engineered systems and into the environment. Some of them are just products, they're in products that we don't know or haven't identified. And so it's hard for us to track them down, actually. We're always playing catch up.
The people who are producing the products don't know that they're putting PFAS in their products?
In some cases, yes.
Yeah, there are cases of there are PFAS manufacturers that actually make the chemicals, but then there are a lot of companies that use products that Maine is running into this issue right now where they created a PFAS ban on various products. And they sort of keep kicking out the enforcement date because a lot of the companies are trying to report, but they don't know how to. They don't know how to run down in their supply chain if they are or not using a PFAS product. So it's creating a challenge when these companies are supposed to report and they suspect they might be using it, but they can't definitively say which one it is or where. So it's an issue, and it's one of the things, if we could get these all out of commerce, that would really reduce the burden on our waste systems, on all the environmental streams. I know we have proposed legislation right now in the New Hampshire legislature that's aimed at a targeted product ban to try to reduce the amount of PFAS coming in. It's currently going through committees and being reviewed, but there are a lot of interstate groups that are trying to work on this issue of trying to figure out how can we either get things out or at least make it easier for companies to recognize it. This is also something that we're hoping at the federal level, there is more action around this. The EPA has gone through with some recent regulatory actions that are full of acronyms that bore most people to tears, but then we get excited about at the state, where they have improved some reporting requirements now for companies, that it is going to have some trickle-down effect on which companies are going to be more aware of what they're using with the reporting requirements. But we're still probably going to be wrestling with this for a while.
To those people ought to know that they're putting PFAS in their product.
They do, and I think 3M earlier this year made an announcement that they're one of the earliest manufacturers. They very proudly held the patent until they got all the news and press for it. So they've announced that they're going to be phasing out of that industry, but there's still a lot of companies in India, China, and other countries that manufacture PFAS, put it in products or reagents or chemicals that we import to the United States and use for manufacturing. So it is not a satisfying answer. It is just more of a grinding your teeth answer, the more even saying it up here. It's frustrating.
Likely have PFAS that you might not want to use.
Yeah.
There is some great nonprofits that have resources that tell you, like the Environmental Working Group is one. They have an app. I don't know if anyone's ever used it, but you can, like, when you're shopping for makeup, for example, they'll give you a whole list of products and tell you what's in it and rank it for you. So there's some good non-governmental resources out there that people are trying to do to help consumers, if that's helpful.
Yeah.
The environmental.
Yeah.
The only one I know of.
Then there was a question over here. I don't want to miss anyone.
Great Bay had an astronomical meaning at the top. It was in red,
Yeah,
which is much higher than even urban China.
yeah, there was, we've taken 47 precipitation samples. And then one of those samples that we collected had a detection of, because we have to touch on, it was one sample. And that's the number that we detected. And one of the things that we're hoping to, so it's not a consistent detection. The rest of those samples were the lower detection. And it's possible that that could have happened, during a time when we had, for some reason, a lot of dirt movement or dust that was coming up, or a storm coming in from a different direction. So we're, the student that's working on this, and this data has just been coming out over the last couple of months, is going to start looking at the storm trajectories for some of these samples and the storm intensities to see if there's something about the way that the precipitation is moving that influences what we're detecting through time. We're also going to be we're also collecting samples of snow to see if we have, differences in snowfall as opposed to rainfall. So it's an ongoing study, but I really appreciate that you saw that red number there and pointed it out. And I should have highlighted it when I was talking. Thank you.
One follow-up to that, is there, when you guys are looking at that precipitation and when you get something that triggers really high that you haven't seen before, are you looking at any, particularly with respect to Great Bay, any of the air traffic coming out of Pease Air Force Base or former Pease Air Force Base?
Oh man, that's such interesting. I didn't know. We haven't even thought about that, and I don't know how we even incorporate it. In fact, just getting the storm trajectory information and how it relates to our sample and where those storms are moving from has been a little bit of a challenge too. So we're mapping, how that precipitation is moving over this regional area. But that's a great idea, and I'll add that to the list of Catherine to look at and see if we can include the air traffic piece and things. Yeah.
The question for you, about the EPA's new MCL, I believe, for drinking water, which is coming out soon now. I believe it's 4 parts per billion or something, which, as I understand it, is near the detection limit for what needs to be detected. And you mentioned some of the rainwater samples were also near that limit. I didn't get to see the numbers. Does that mean that the rainwater itself at this point is near the EPA's upcoming MCLs, or am I off?
The MCLs are 4 parts per trillion. So they're, and a lot of the methods that are being used right now have a 1 part per trillion detection limit.
Okay.
So many of the values that we're reporting in precipitation are right around or below that. And the analytical labs, they report these as kind of an estimated. Okay, so we're really barely detecting in a lot of cases anything in the precipitation, but enough for us to be able to record those values consistently and feel confident about what we're seeing. Yeah.
There's where your storm patterns are going to be really important.
Absolutely. Yeah, we did have a couple of, so I mean, there were statistical differences, but we are seeing some different concentrations at some of the farming areas. And, you know, that's where we have a lot of movement of some of these particles. And so that's something I think that's really important on the terrestrial side.
I have a question about the City of Dover project. So why the City of Dover? Were they like the first ones? Were other towns contacted and not interested? And will you consider, you know, is there opportunities for in the future for towns to participate in a project like that?
Thank you. So every year we have a number of capstone projects that are proposed for our senior engineering students. And I have worked with the City of Dover on previous projects. And it actually takes quite a lot of effort and commitment for somebody to sign on to one of these projects. We advise the students on a weekly basis. And so it has to take an interested party to submit a project. The students then rank the topics that they're interested in. They get assigned an advisor like myself. And then we work with the sponsors and advisors and students through the year. And in this case, the City of Dover had some money that they were interested in putting towards this project that came out of their budget. And that money drove the number of samples that we were able to collect. And so there was a lot of pieces that came together to make this happen. But I will say that we are always looking to support communities in these projects. And there's funding, I think, that can be tapped through different state agency, like source water protection funds, for example, to try to understand within a community where maybe some of these other sources might be. And we have students that are always very interested in assisting with these projects, especially if we can send those samples to an analytical lab that has a standard method. We can help collect and analyze the data. We're very good at that. So if we could help out with that at a community level, we'd be happy to.
Good to know. Thank you.
Yeah.
Hey, John.
Oh, yep.
I just want to let you know there's someone with their hand raised online, just so you know.
Sure. Can we come back to you really fast and let this person? Yeah. So let's go and go to the person online.
All right.
I'll
I'll try.
also mention briefly that there are a couple of questions in the chat too.
Yep.
Oh. Okay.
Mark M, I'm gonna unmute you. You might have to unmute yourself. Mark? Why don't we try the chat questions first? And Mark, if you're having trouble with your audio, you can always type into the chat too.
Okay. So Rob, I'll let you read off some of the questions.
Sure, and there's just 2 so far. First one's for Jonathan. Is expected portion size the only reason why shellfish and finfish have different screening levels?
Yes. So the big difference between their screening levels is the amount of finfish versus shellfish. So typically people eat less shellfish than they do finfish. But this is one of the useful things about the work that Dr. Crawford did in that paper is we want to be able to go back and look at our own screening levels when that paper is finalized, since we now know and can show that New Hampshire residents have higher fish consumption patterns and seafood consumption patterns. That doesn't only affect our screening levels, but we're also going to be looking at updating a lot of our fish consumption advisories in the state. So outside of what we do here at the Great Bay and in the estuary, we actually have advisories for other contaminants across the state. Some spots for PFAS, everywhere for mercury. But we're going to be looking at updating those advisories. And that's, again, really unique aspect of this research where it's not only helping us with PFAS, it's helping us with other contaminants in other parts of the state. And we really value having research like that where we get a lot of mileage out of it.
Great, thanks. One more. This one's for Dr. Mauser. It says, I think on slide 15, where you're showing the median values in precipitation that were reported, do you have a table with the high end, the 90th or the 95th percentile, or the highest values, or know the data offhand for PFAS? Thank you.
Thanks. I
Helpful.
believe we had, thank you. Right, we're reporting the median concentrations, and there were a couple, and when I say a couple, I mean just a few that were above the proposed drinking water limits. And I can go back and look at that more specifically. Again, when we saw a couple of samples when we first started analyzing the precipitation, and we weren't sure exactly if it was related to the way that the storms were moving or the type of a storm, but consistently we're seeing really low concentrations. It doesn't seem to be something that's happening regularly. But you're right. We need to report that data with the statistical range so that we are more transparent with the total values that we're seeing. Thank you.
And did Mark write a question in, or do we have Mark with us now?
He's still here. There's no question in the chat.
Okay, we'll take a question from the back of the room then.
Your team for doing such a fantastic job pulling all this together. I am curious if there's another team like you guys in the West Coast or in some other university or whatever, or are you guys breaking ground on this information? Number one. Number two, we've been focusing on Great Bay, local here. Is great. Do we have more data that says that Great Bay is an outlier compared to, say, up and down the East Coast, Chesapeake Bay, or something up and down on the West Coast or outside of the United States? I know Chris touched on that on an earlier question, but just curious what you might have to say to that.
So it's like asking someone to pick their favorite child. I have to be careful what I say next. So with New Hampshire, what we have here that's very unique is we have an estuary system, that we have an AFFF site on the estuary system, but we also have a lot of wastewater plants, non-point sources, and other things coming together. That's actually not too unique for a lot of other coastal spots along the eastern seaboard and even on the western seaboard. What is unique here is the sort of setup that we have of having researchers and regulators and folks that are very familiar with that system, that have been able to leverage a lot of resources to ask these questions here. So one of the programs that is funding Dr. Chen's work is through the Department of Defense. It is not a site investigation group. So it's not like when you go to a meeting and it's a RAB and they're saying, this is what we sampled and that's it. This is a specific program for trying to actually understand these broader scientific questions, because it can help answer questions at other sites. It's incredibly useful to us here, but right, it's that mileage thing of what's helpful to others. So we're just really lucky that we have a bunch of folks that before working on PFAS worked in a lot of other contaminants and have been able to apply that knowledge here. There are other research teams in other parts of the country asking slightly different questions based on what information they have and who's taking a part in that project. So there are those groups. Some of that funding comes from Sea Grant, which New Hampshire has a Sea Grant program that has funded some of this work to actually look at these PFAS questions. But there are other national Sea Grant programs that are asking other questions about PFAS in those sites that we're looking forward to learn from. They actually had a very large application cycle about, was it a year and a half ago, 2 years ago? I feel like time flies when COVID annihilates your memory. But it was specifically targeted. It was a large sum of money targeted PFAS. A lot of those projects are going on right now, and we're waiting to get their results as well. So there is other movement in other parts of the country that we're looking forward to.
Just hopefully some of the work that you're doing collaborates with them and can really getting a bigger picture of what's going on. And we also wondered is, are the numbers that we're seeing here locally, an outlier compared to the rest of the different places?
I wouldn't say so. There are some AFFF sites that they were still discharging, still using it, and because of hydrology, because of geology, because of the setup, there are some places that are much, much higher. It doesn't diminish what we're seeing here whatsoever, and that's one of the reasons why we're still doing the research and putting time into this, is to understand it. But really, we're sort of at the stage where probably a lot of sites might be in the future, where they're still trying to figure out what these lower but still detectable levels are, but also how do you tease that apart from all these other sources until we can get a handle on this problem with our waste systems and PFAS coming in. Because to be frank, once we're out in the bay, it's hard to really tell if it's whether or not it's AFFF, if it's non-point source runoff, if someone just recently waxed their boat, what it could be. So it's a challenge for us, but that's one of the things we're still trying to tease apart.
John, can I just jump in? I just wanted to also mention that there's a couple Superfund research programs, center programs in the US who have focused their entire programs, and this is like, 20 or 30 researchers in a center on PFAS. So down in North Carolina State has one, and they focus on the Cape Fear River. So there are other places where they have a lot of research going on around specific contaminated sites. The University of Rhode Island Superfund research program focuses on Otis Air Force Base in Cape Cod. And also I should say that the Department of Defense, which has used AFFF a lot, is funding over 300 projects right now on PFAS. So all over the country, because there's plenty of sites, to look at, many of them military sites.
Are really, I think, in terms of the work that the DES has done and taking samples and characterizing our state as a whole. When I look at what other states have done and the data that's available, New Hampshire has a lot of information to draw from and is leading the way in some of these in the thinking of where the sources are and where the public is are. And so I think we should really thank them for all of their efforts. We also are, as researchers, our study areas are driven by where the problems are and where the funding is. We would love to be able to expand our work beyond the Great Bay because we know there are a lot of potential issues in the state. And so we're always looking for ways to fund that research, even through these federal funding agencies, through appropriations, through state funding. So we're willing to address any area if we are able to fund the need.
Yep, Andrew.
Here, I understand something you said earlier about fish advisories. Are there any fish advisories in the state for PFAS?
In the areas of southern New Hampshire, basically in that southern area south of Merrimack, going down to about Nashua, Salem, in that triangle area, we do have fish consumption advisories in 5 of 14 lakes that we were able to sample for fish. That sampling was done in 2000, which was really interesting to get people to go out and sampling when we were still like very COVID precautious. So really interesting game of get the fish. But we had one-time funding. We were able to go out and sample some of those lakes. We sampled those based on highly populated areas. They're frequently accessed by a lot of people, but because of the large PFAS contamination area down there, we sampled those. And since they're lakes, we don't have the transient issue of which fish are we sampling, which time of the year. There were 5 lakes that there's sufficient PFAS, specifically PFOS concentrations, that we issued fish consumption advisories that are more restrictive than what we already have for mercury across the state. We've gone to the legislature several times and explained if we have more funding, we can go out and sample more fish. we don't have an instrument at the state for analyzing fish, shellfish, or any of those items for PFAS. Any of the state testing that's been done, we have to pay a contract lab, specifically one in Canada, that it's expensive to send stuff to, and now they're actually not taking samples. So again, that's where either having these partnerships. So with Dr. Chen's group, they are working with Clarkson University out of New York. They've been great partners. We would like to get more of that capability set up in the state. The state is looking at setting up an instrument to analyze drinking water samples. But once you have a drinking water machine, nothing else can go on it. You can't test sediment, soil, nothing else. You just leave it alone. It is a magic machine. So that is something that for like my program, the environmental health program, looking at fish, shellfish, deer, other things, it would be really helpful to have that, but that's just not something that is within the state budget to support. We would require technician time for that. We would need dedicated space for another instrument, and we just don't have that at this time. So
a follow-up to that, in terms of no fish advisories being set for this area, is it because you don't have enough data to support it yet, or the data that you do have indicates that that's not needed?
we do not have the data here for setting advisories within the Great Bay. We would need to go out and we run into the issue of we need to sample the species. We need to make sure that there are species. We know which ones are transient, which ones are here, which are representative, but we don't have the funding to do that kind of sampling.
So when people ask in the community, like, should I be eating fish or shellfish from the Great Bay? Like, if they're concerned about PFAS levels, how do you answer that question? How would you answer that question?
Bluntly, based on the work that we have been doing with folks, oh, I see hand went up. So we'll come back to Mark because I think his hand went up.
Okay, I think
so based on the work,
got unmuted here. It was a crash, okay. And I also found I was blocked out of chatting because it said I needed to connect to somebody to directly chat. So I don't know what the hell I was doing, but I should have just come in person because I'm in Portsmouth. But anyways, I have 3 questions if I can, or you can finish what you were just talking about and then I'll give them.
let me quickly finish what I was just talking about, and then I will call on you. So the question is someone's asking if fish in the Great Bay are safe to consume. At this point, we don't have data to know what the concentrations are in them. Based on what we're seeing from the broader offshore fish, we know fish can be a source of PFAS for people. So if people are concerned about their exposure, we recommend limiting the fish consumption, but without further data, it's hard to say that it's high enough that it's a should not eat. The other challenge that we run into is a lot of times for coastal fish, we're really getting into the area of that is FDA's jurisdiction. And I will be perfectly blunt, and a lawyer will probably drop from the ceiling and get me, but the FDA is not doing a lot right now on addressing that question. They have looked at broader market basket samples, but they're still sort of behind in catching up to where some of the states are in trying to sample this. So it is something we like to look into. We know that there are other groups looking at Gulf of Maine fish. Some folks are looking at migratory fish and these studies that we mentioned before that are ongoing. If we can find ways to start relating that to water concentrations, that opens up some avenues for assessment. But really the big thing that would be helpful is getting the support and resources for us to go out and collect some of these samples and actually know. So with that, we're going to go to Mark. You have 3 questions and you've been incredibly patient, and thank you for not just driving here and wringing our neck for using Teams.
Well, I probably could have driven there in the time I spent trying to get connected one way or another. But,
sure.
But first of all, I just wanted to thank you for this presentation because I've been on the RAB for my 3rd term now, and this is what I've been asking about for all 3 terms. So thank you very much. More detailed questions perhaps, but one for Dr. Crawford is, look, I'm a forager, all right? And it seems to me you could take a look at the number of shellfish licenses and issued by the state to get an idea of a subpopulation exposed to foraging shellfish out of Great Bay. And then link their risk to the data that you have from Great Bay on shellfish contamination, specifically with respect to soft shell clams, oysters, and mussels? Is that something you could do down the road?
Yeah, Mark, thanks. That's a really great question. And that is actually, well, broadly speaking, looking at populations of people who consume seafood in a manner that is not this kind of typical general New Hampshire population type of person is exactly an area of research that we're hoping to expand into next. So you raise a really great point, and it's definitely something on our radar. And I think that your idea to use fishing and shellfishing licenses as a way to investigate a foraging population is a great one, and one that we'll certainly take into consideration as we do move forward with some of our next steps. But yeah, thanks for raising that point.
Yeah, good. That's good. Thank you. And then these other two are for Dr. Chen. With regard to bivalve species, I find it fascinating the difference of pattern of PCB percentages by species, which in my opinion reflect different foraging strategies, or perhaps different spatial distributions, or both. The question is, within a bivalve species, have you looked or will you look at age or size specific concentrations to test if bioaccumulation accumulates in older, i.e. larger, i.e. most harvestable members of the same species?
Yeah, great question, Mark, about the size effect. You know, it's been interesting reading the literature on PFAS, because normally you think, oh, older and bigger fish actually have higher concentrations. That hasn't been the case in most studies for PFAS. Older and bigger fish don't necessarily have higher concentrations. We do have a range of sizes of individuals, like especially ones where they were really abundant, blue mussels. So within that species, within a site, we could look at a size range and see if there's any difference. But we haven't done that yet. We have the ability to, because we, you know, when we went out for that 2022 sampling, we did collect a range of sizes. And we have wondered about that, because we always see that with mercury. You know, it's always higher concentrations in bigger, older individuals. Not actually so much with invertebrates, so I would say, mostly with fish. But that's not to say it wouldn't be the case with these bivalves. Now, the ones that we, I showed you data for, are mostly, you know, the size that people consume. We actually, and I should have thanked Chris Nash of the shellfish program at New Hampshire DES. He really took us to the places that they go and collect. Some of the places are closed, as John mentioned earlier, but others are where people do, you know, go to get their shellfish. So the size question is a good one. And ecologically, it would be really interesting, but I do find it surprising that so far I see there isn't much of that pattern in fish.
Yeah, and the other reason, well, I was asking about shellfish,
Yeah.
but also fish, because that's where my insights come from. But because, for example, if you take a composite sample of different size classes, and in fact there are differences in concentrations by size, then you've essentially masked the differences.
Totally, yeah. No, when we collect our samples, we are categorizing them by size range.
Great.
Yeah, absolutely, because you're absolutely right, because it could confound the, you know, the concentrations. Yeah.
Exactly. You know, an unknown classification variable.
Absolutely.
All right.
Yeah.
Second question for you is, I view most of the bivalves you've looked at, I don't know much about razor clams, as primarily particulate particle feeders.
Mm-hmm.
And they're actually size selective particle feeders, size selective by age, size selective by, shall I say, mesh size. And was wondering if you looked at seeing the difference in percentages of different PFAS among the different species, and recognizing that they might have different foraging strategies and different size selectivity. It seems it would be valuable to fractionate the particulate material, i.e. zooplankton and phytoplankton, in a whole water sample to match up with the size range reported for feeding of each of these species of bivalves, and see if you get a better fit between the PFAS mix in the size fraction, the particle size fraction of the water column, and what's in the shellfish.
Yeah, you actually have just articulated what we have tried to do. I can tell you that we do that more easily in a lake because in the estuary, if you don't hit it exactly at slack tide, it's really hard to get a good particulate sample. There's just so much debris and detritus. We have tried, and I agree with you that, like, say for the oysters and the blue mussels, that they are definitely pulling from the water column, and the particle size and what the particles actually are. Are they detrital? Are they actually, phytoplankton or zooplankton? Yeah, you should come up to our lab. We could definitely talk more about this.
I'd love to. I was going to make a trip up. You had a presentation maybe 6 months ago.
Oh, yeah, I think so. We've done a bunch of them. Yeah, so I just wanted to say that for mercury, we do see a very strong relationship between fish, like forage fish, and particulate concentration. So particulates can be really important. It's just not that easy to get them in the estuary, I have to say.
Okay, yeah, because my insight comes from PCBs and mercury.
Yeah, I'm hearing you.
Okay, so I'm just gonna jump in here. So really quick, I just want to be mindful of time. It is 5 after, and we said this is going to go until 8 o'clock. So one, I just wanted to thank everyone for coming out here tonight. And I also want to thank Dr. Mauser, Dr. Chen, and Dr. Crawford for giving us their time. I'm going to stick around for a little bit. I'm going to leave the video conference going. I realize if folks want to get going because it's 8 o'clock on a Thursday night, and it's been a long week for me, so I imagine it's a long week for everyone else. Again, just thank you all for coming out, and we will come back next Thursday with a different group to talk about more terrestrial and backyard issues and less about seafood and aquatic issues. So thank you.
Thanks very much. Thank you so much.
Do you want us to stay, John, or no?
Really quick, are there any other questions for Dr. Chen or Dr. Crawford in the room? Okay, no hands are going up. People are getting up. Is there anyone else in the chat that has a question for Dr. Chen or Dr. Crawford? Okay, no, if not, I think you two are free. Rob, is there any other questions for me or Dr. Mauser?
Looks like just thank yous.
Okay, terrific.
But I'll stop recording.
Thanks, everyone.
Yep. Hey, Rob, how many attendees were there?
I can tell you after, because everybody leaves.
Okay, sounds good. Thank you.
All right,
Bye, John.
Bye.